The book is called The State of Science, and an early version of the book was called Good Science, Bad Science, New Science, Old Science, sort of a play on Dr. Seuss. And so what I want to do today is talk a little bit about one example of old science, and that's something called fluorescent proteins. And then 3 examples of new science. So I want to talk a little bit about optogenetics, which is sort of, I guess, in a science fiction-y sort of way, a bit like mind control. And then the other 2 are CRISPR, which is gene editing, and deep learning. And so I want to talk about those methods, but there are some sort of deeper questions that I want you to think about when I talk about them. And that is really, should there be a limit to science? Is science getting a little too big? Should there be some controls on science? And then I also just want to use these, they're really techniques as examples to show that applied science is really important. So science, not applied, sorry, basic science. Science that you're doing just out of pure interest, that you're not doing to get money out of it, you're not doing it because you think there'll be a technology coming out of it, but you want to understand something. And there doesn't seem to be an obvious use. So if we're talking about the size of science, I went to school in South Africa, the University of Witwatersrand, and one of the first classes I took was a zoology class. And in this zoology class, professor introduced this essay by Jack Haldane called On Being the Right Size. It's from 1926, so it's really old, but for some reason I remember it well. And in this paper, he says that if you take a mouse and you drop it from a high building, it'll probably survive. If you take a human, they'll probably die. If you take a horse, they'll splatter. It's rather graphic, but it sort of shows you the importance of size and gravity. And then he went on to say that if you have a small animal like a locust, the locust doesn't need big heart and lungs to push blood all around its body because the air can diffuse pretty much everywhere. But if you have a big animal like an elephant and the blood needs to get all the way to its trunk and all the way to its hind limbs, it needs a really big heart, really big lungs. It gets really hot, it needs a large amount of surface area to cool down. But the problem is that surface area increases a lot slower than volume. So as an animal gets bigger and bigger, there's less surface area to cool it down. It gets more and more complicated. And so he presented in 1926 a series of formulas by which you could calculate what was the ideal size of an animal and what was the maximum. And basically an elephant is the maximum size of an animal that you can get on Earth. Its heart is really big, its lungs are really big, its bones have to be very thick to support all that mass, and it has to eat all the time to get energy to survive. Anything bigger wouldn't work. Probably if I gave this class or this talk at an elementary school, there'd be some elementary school kids saying, whoa, 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 what about whales? Yeah, whales get bigger than elephants, but that's because they're in the water, which is buoyant and which counteracts gravity. And whales are about as big as you're going to get in the water. And this being the right size probably also works for mechanical things. You think about the mechanical things around us, an aeroplane, a car, a ship. There's a maximum size. Make it any bigger, it's going to be really difficult and very cumbersome for it to move, to fly, to use a lot of fuel. There's an ideal size and there's a maximum size. Maybe even with government and corporations, Google, and if it gets too big, it gets so complicated that something can go wrong in one part of Google that nobody even thought about. It'll affect the whole company. So is that true with science? Is there a right size for science? Should we think about it, it turns out that science is growing really fast, faster and faster and faster. These techniques that I'm going to tell you about didn't exist in 2010, and there are tens of thousands of people using them every day all over the world. So that's really fast growth. They're incredibly potent, and the more powerful they are, the more risk there's associated with them. So I just want you to think back to these questions as I tell you about these interesting new techniques. I'll start with what I do, just because it's interesting and it's actually related to the new techniques I'm going to tell you about. So I'm old, and this is an old technique. So I use computers to calculate how fluorescent proteins give off light. Fluorescence is if you shine a light with high energy on something, it takes that energy and immediately converts it to a color that's lower energy. So here you can see some corals, and these corals have white light shining on them. If I change it to blue light, you see the blue light is absorbed and the coral gives off a yellow light. It turns out in water, blue light travels further than any other color. So all the marine organisms all react to blue light because it just travels the furthest. So I'm interested in these type of proteins. I'm particularly interested in one that's called green fluorescent protein. This is found in a jellyfish. And what we have here is the genetic sequence, the gene for this protein. You can think of it as the recipe on how to make green fluorescent protein. On top there, you can see A, T, G, A. Those are the nucleotide bases. Beneath it are like the words of the recipe. These are the letters. These are the words. They're the amino acids. And so this is basically the recipe how to make green fluorescent protein. There's just one extra thing that happens with proteins. If you make this recipe in any organism, in a butterfly, in a cotton plant, in a bird, if they make this gene, this protein, then it will fold in exactly the same way. So it's like having a recipe. If you've got a certain recipe, it always folds in a certain way. It turns out that GFP always folds into this soda can shape. And if you shine a blue light at this, it's called green fluorescent protein. It gives off green light. So I calculate, if you go here, there are 238 little amino acids. If I change, say, this Y to an H, what happens? It turns out if you do that, then you get a blue fluorescent protein. So I use computers to calculate how that would change the three-dimensional shape. We know from experiments that it has this shape, but it's very expensive and difficult to do the experiments to determine the shape. So with a computer, I can just go and say, oh, I'm going to change just this little part, see what happens. So why would I want to do this? Well, these proteins are incredibly useful. So here we have DNA. This is just another way of writing the recipe, the gene. So this is the recipe to make this protein. And just like a recipe book, this recipe will make this protein and no other protein, right? So if you've got a recipe to make a raspberry cake, it'll always make a raspberry cake, not a pecan pie or anything like that, no matter who makes it, no matter where they make it. There might be some differences, but that's really because of the way you make it. At the beginning, there's going to be something that says, hey, this is the raspberry cake recipe. At the end, there's going to be something that says, stop, you're done. And then it'll make this protein. Well, what if you went and you edited the recipe book? And so at the end of the recipe for the raspberry cake, you put an extra recipe that tells you how to make extra icing on top of it. So then whenever anything makes the cake in any place, they'll always make the raspberry cake with extra icing on top of it. So we can do the same thing here. We can take the gene for this protein, the recipe, and at the end here, instead of a gene for icing, we have the gene, the recipe, how to make green fluorescent protein. So whenever this is made, hanging off it will be like a tiny little light bulb. And you can shine a blue light on it, and you can see where it goes to. So let me show you a use of this. This is from a couple of months ago. So what these researchers have done, so I didn't do this, but they took the virus that's responsible for COVID, and it actually has in it 39 genes that code for 39 proteins. You can take one of them and genetically modify it so it has red fluorescent protein on it. So now you can see individual viruses. These individual viruses in this case have infected the bronchial tubes of a ferret. It turns out ferrets are the best model systems for humans if you're studying respiratory diseases. So if you're studying the flu, typically people use a ferret. If you study COVID, they use ferrets. Now what you can see here is in all these places where there's red, these are places where the virus has attacked the cells and has multiplied. So you can see by how bright it is, how many you have. And so you can follow exactly how the infection goes. So that's why this fluorescent protein is very, very useful. Now, there are two reasons I started just telling you a bit about my research, and you'll see why in a little bit. It turns out optogenetics is based on fluorescent proteins. And it also turns out that the type of calculations I do are going to be calculations that can also be done by machine learning, two of the other new techniques. Now, this is an old technique. In 2008, the people who did this work got a Nobel Prize for it. In 2014, people who improved it also got a Nobel Prize for it. So this is sort of an old science, but you can see it came from basic science. There was a Japanese researcher who worked at Princeton who caught over 20 years about 1 million jellyfish and studied those jellyfish just to find out how this green fluorescent that's now been modified to be red fluorescent protein worked in a jellyfish. He didn't want to use it for anything. He just wanted to understand how it gave off light and why it gave off light. And then only in the late 90s did people start thinking about, oh, we can use this, we call it a tracer, as a tracer, where you tie green fluorescent protein to a protein you're interested in, and you can see where it goes to. So you can do this, for example, with a cancer cell. You can make a cancer cell fluorescent, and you can implant it into a mouse. If it multiplies, you'll see the fluorescence increase. If it starts metastasizing, moving through the lymphatic system or the blood system, you can actually see individual cancer cells moving through the blood system. So it's a very useful sort of tag. So let's go on to a much more powerful method. I think if you think of fluorescent proteins, you can think of it as a microscope. So this is something called optogenetics. Opto meaning light, and genetics meaning we've genetically modified something. So I want you to sort of to sit where you are and do an exercise for me. Sit and think of your big toe and twiddle your big toe. Move it up and down. When you're doing that, there's a message that goes from your brain to your big toe, and the muscles contract and your toe moves. How does that work? Well, it turns out that you've got neurons that go from your brain all the way to your toe, but there's not just one neuron, there's a whole series of neurons. And what happens is, if you think of this, my cursor being one neuron here, a neuron gets the signal that's going to go to your toe. The signal sends a chemical signal to the next neuron that says, okay, go and fire. And when it sends it to here, the amount of positive ions increases. If it goes over a certain amount, then this neuron sends the signal to its end that says, okay, go and twitch that toe. It goes to the end and then to the next one, and to the next one, to the next one, till it gets to the muscle, and then it contracts. Now, 10 years ago, if you wanted to see how neurons worked, the only way you could do this was by taking a very, very fine needle, an electrode, putting it wherever you're interested in the brain or something, and applying a very fine current. But when you did this, you'd excite thousands of neurons. But we'd really want to excite just one of them and see what happens. So can I make just this one fire? So the mouse has nothing to do with this, if it's in a mouse, for example. I want to make just this one neuron fire, and I want to see what happens. So this is where we go back to basic science again. Algae. Algae are singular cellular organisms. They're like little balls. They float in the water. They get their energy from photosynthesis. They're green. So if you have an algae and you have a completely dark aquarium, the algae will just randomly float around the aquarium. But if you turn on a blue light anywhere on the algae, anywhere around the aquarium, the algae will actually swim towards the blue light. So that's what you can see in this experiment on top here. Here's a blue light, here's a white light. It swims to the blue light, again, because blue light travels the furthest in water. But how does it do it? An algae doesn't have eyes. How does it know where the light's coming from? How does it know where the sun is? It's not gravity, because you can put the light on the side. You can even put it at the bottom. If you put it from the bottom of the aquarium, they'll all swim to the bottom. So they can see the light. And so how do they get there? Well, it turns out that the algae, you can think of it like a soccer ball. The soccer ball is studded with these things called channel rhodopsin. There are thousands of them all over this algae. And if the sun's on top, the sun's shining on top here. And so the sun hits all of these little channels that are on the top of the algae. When the blue light hits them, they open up. When they open up, they suck in all the positive ions that go in here. And so all these positive ions go in. So remember what I had said back here, that when a neuron fires, it gets a chemical signal and all the positive ions go into the neurons. And once it reaches a certain concentration, this fires. So what if we took these channel rhodopsin from the algae and put them into the neuron? So now imagine this is in a neuron. I shine a light on it, a blue light. When the blue light shines on this, this opens, the sodium goes in there, and that means the neuron will fire. So now we can genetically modify neurons anywhere in the brain, in the heart, on a muscle, and we can use a blue laser that's very, very fine, shine it on that neuron, it'll open and it'll fire. So now we could take individual neurons and make them fire. So let's just see how you could do this. This is sort of a 6-step program, how you could do this. Here's the gene, the recipe for this channel rhodopsin. So this is what was found in the algae. And here's this sort of thing that says, hey, go and make the recipe. So you take this and you put it into a virus, a harmless virus. You inject the virus into the brain of the mouse. The virus then takes the genetic material and inserts it into the genetic material in the brain of the mouse. So now your mouse has this one algae protein in here. If you could shine a blue light on it, it would fire. But of course, the brain has a thick skull around it. So what you need to do is you make a small little hole, pinprick size hole, and you take an optic fiber that can shine blue light through that hole onto the brain. And so it shines it on the brain, on the neuron. When the light hits here, the neuron opens and that neuron fires. Now you can genetically modify a mouse, for example, so that only the neurons that go from the whiskers to the cerebral cortex at the back of the brain are modified. So you can activate those just by themselves and none others. Here's an example. This was on the cover of Science magazine of a mouse that is optogenetically controlled. It's one of the first ones. And so what was done here was that just the right-hand side of the brain was genetically modified. So if you remember, in a brain, the right-hand side controls the left-hand side of the body. So in this mouse, what you have is the mouse is being genetically modified. So the neurons on the right-hand side get turned on if you shine a light on it. And this gives the mouse the urge to run and turn to the left. Oops. So here what you'll see is the mouse. If you look on top there, that's the optic fiber. When it turns on, when it turns blue, the mouse will run, turn to the left, running, turning to the left, turning to the left. Switch it off and the mouse will stop. And it's quite happy. It's not excessively grooming or hiding in a corner or anything like that. And then if you turned on the light again, it would run and turn to the left. So you can see this is a very simple way of doing this. So coming from research on algae, what we've got is this really cool method that we can start seeing what happens if you activate individual neurons. There are lots of research uses, but there are also some medical uses. This is one that's going on right now in Texas in the clinic, in Texas Southwestern University. This is normally what a retina looks like. There are two types of cells, cone cells and rod cells. And lights hits these cells, and then they get activated and they send a message to the optic nerve. It goes to your brain, and depending on which nerves were activated, you see a different color. And depending on where it was, you can see what the shape is. Now, some people have a disease called retinitis pigmentosa. In this disease, the rod cells and the cone cells have collapsed, and light no longer activates them. And so these people are blind. Well, what you can do is you can genetically modify these cells so they have channel rhodopsin in them. If a blue light hits here, it activates the neurons, and the optic nerve can see this. And so far they've operated and genetically modified the eyes of 8 people, and all of them can see enough from being totally blind to be able to see a car coming and to be able to walk with a little bit of help. They can't see colors, but they can see shapes and they can see movements. So you can see the power of this technique. You can also see that this technique might be abused. In this case, maybe not quite yet. This is really interesting. There's a part of the brain, a set of neurons that are responsible for object chasing behavior. They're responsible for a mouse chasing something. So what you're going to see is this mouse, again, genetically modified, so that the neurons that are responsible for the object chasing behavior have here channel rhodopsin in it, and also a fluorescent protein. So the neurons actually light up when they're activated. So you can see that too. But so let's just see how that works. So here's the brain. It's going to be stimulated with a fiber optic cord. And you can see the light is on. When the light is on, this mouse is going to chase the object. Turn the light off and it stops chasing it. It's off. The mouse just sits and ignores it. It'll chase it even if it means jumping off into a smaller object, which a normal mouse or rat will never do. Just, it's so desperate to get this now because the light is on that it jumps down there. It's still looking for it and it'll hang on. If they turned the light off now, it would let go and it would just sit there and ignore everything. It's not going to jump into the water. There are limits. So what can you do with it? This is sort of a crazy thing. So now they've taken an object, pasted it onto the mouse that they can move with remote control. The mouse is going to chase the object. So now you basically got a remote controlled mouse because you can turn it, and whichever way you turn the ball, the mouse is going to go. So we'll see this in just a bit as the mouse goes through a maze. So we have this maze, the three-dimensional maze with all kinds of objects that distract the mouse. There's a zigzag course. It's a male mouse that's distracted by a female. There's a blind alley. There's some bumps that it's got to go over all the way to the top. And you know, a mouse would get distracted by the food, by the female. It wouldn't want to go over the narrow bridge. But now what you can take your mouse that's been optogenetically modified, and it will follow the ball all the way through the maze, ignores the female because it just wants to catch this darn ball, and makes its way all the way through the maze. So you can think of this, it took about 10 years before they managed to get a robot to get to the center of the nuclear core at Fukushima, because there were no small gaps for the robot to get through. So if you had a mouse like this, would you be able to steer the mouse to go somewhere really, really small? And then, of course, now you can see the normal mouse, it's just ignoring everything. So let's just see, has anybody got any questions about optogenetics, about what you can do with optogenetics? I'll tell you some of the things they do that scare me, but let's just first see, has anybody got a question? Just unmute yourself and now we're good? Okay. So there, at Yale, just down the road, there's a group that looks at a gene that's responsible for aggression. And so you can turn on the set of neurons in a mouse and make it aggressive. And there's this test they have where they have a long glass tube. They put a mouse on both ends, and then the mouse have, you know, they both want to get out. And typically what will happen is the more passive mouse will just reverse out, and the more aggressive one will push it out, and out it goes. But if you then genetically modify the passive mouse, you turn on the blue light, and then it'll be aggressive, and it'll push the aggressive mouse out. Switch off the light, and it'll go back again. Now, to me, I can't really see a good use for studying that. Why do we need to know how to make mice more aggressive? Is the next step sort of to make people more aggressive? So that's sort of, to me, the optogenetics has got a ton of good uses, like we saw with treating blindness. It can be used to treat arrhythmia in the heart, all kinds of things. It actually even looks as if in 10, 15 years, we'll be able to treat Parkinson's with optogenetics. So at the moment, there's something called deep brain stimulation, in which a very few cases of Parkinson's disease, they have an electrode inserted. It's called deep brain because it's really deep in the brain. And when they have really bad tremors, they can push a button that then shocks their own brain, a very small micro shock, very deep, and the tremors stop. Now, it turns out that you can actually get to that part of the brain optogenetically much, much easier. And of course, a light is A, more accurate, and B, less damaging. So the same idea would be if somebody has Parkinson's disease, they'll now have something that they can push a button, turns on a blue light, the blue light stops the tremors. And so they're working on it. So there are a lot of good things you can do, but there are also a lot of really scary things. Well, where you can control what animals and people are doing. So that's quite scary. A couple of weeks ago was the announcement of the Nobel Prizes, and the Nobel Prize in Chemistry was awarded at 5 in the morning. I woke up to watch it, and this is the announcement.
Sciences has today decided to award the 2020 Nobel Prize in Chemistry jointly to Emmanuel Charpentier and Jennifer Doudna for the development of a method for genome editing.
So I thought this was really cool. In fact, I had been expecting this, and I'd already written an article for a newspaper. So when I heard this, I had to change it a little bit because I thought there would be a third person getting it, and then sent it off. I also have a chapter about Jennifer Doudna and Emmanuel Charpentier and CRISPR, which is the technique that they got the prize for. And so I want to tell you a little bit about this technique. So after the prize was announced, the chairman of the Chemistry Nobel Prize Committee, that's the group that actually decided that these two women should get the Nobel Prize, came and gave a description of why they got the prize.
The Royal Swedish Academy of Science.
And here it is.
The enormous power of this technology, it means that we need to use it with great care. But it's equally clear that this is a technology, a method that will provide humankind with great opportunities.
So the Nobel Prize came with a warning label, right? He said, hey, this is a really powerful technique, but we got to watch out. You know, people can do crazy stuff with this technique. And we shouldn't just go into it completely blind. Now, he wasn't the first one to warn us. In February 2016, James Clapper, who was the US Director of National Intelligence under Obama, every year the National Intelligence Director has to give a speech in which he talks about worldwide threats to the US. And in his speech, he listed 6 things that we have to be careful of. Most of them were obvious ones. It was that North Korea might develop weapons of mass destruction, that China is modernizing its nuclear forces, that Iraq and Syria are using chemical weapons. And then there was this 6th one, CRISPR, genetically engineering, and that is going to be something that we have to be really careful of. So it's really interesting, right, that this is the National Intelligence Director who's saying this. At the same time, the director of the FDA gave a talk, FDA's Food and Drug Administration, and he said CRISPR, this gene editing tool, is the most fascinating and coolest development we've seen yet. It's going to change medicine in ways we can't believe. And we're going to get tens of thousands of new medicines based on CRISPR. So we have this technique that's really, really cool, but is also quite dangerous. So let me tell you a little bit about it. So CRISPR is a gene editing tool. 10 years ago, if you wanted to go and you wanted to edit, say, a ferret cell, and so you've got a cell from a ferret and you want to go and put in that GFP gene that I was telling you about into the ferret cell. And nobody's really worked on that type of cell before. It would have probably cost tens of thousands of dollars, maybe even hundreds of thousands of dollars, and would have taken years for you to get to that point where you can insert the gene exactly where you want, the way you want it. So when we talk about editing, it's as if we were back in the days where everybody was typing with a typewriter. So imagine you're using a typewriter, you've written your whole book, and at the end of the book, you realize that you've spelled somebody's name wrong. I spelled Bethany's name with an E instead of an A. And so now I've got to go back and find her name, got to read through, find it, change it. Fortunately, I only made a mistake with an E and A, so it doesn't change the spacing, but still it's really, really difficult. That's how gene editing was before CRISPR came along. CRISPR is like a word. It's a computer editing program. You can now go and find any sort of genetic material, change it very quickly. You can take something out, replace it. You can actually take it and say, okay, leave it there, but just instead of making GFP, make more GFP or make less GFP. So it's a very, very good word processing technique that just edits genes. So now, if you wanted to edit a new cell that's never been edited before from a new species, it'll take less than a week and it'll cost less than $100. And you get kits that you buy, you go online and you actually type the DNA sequence that you want, and you type the one that you want to replace it with, and you pay your $100, and FedEx sends it to you 3 or 4 days later, and you get the whole CRISPR DNA unit and instructions on how to use it. And so now you can genetically modify anything. So that means research is a lot easier, right? But it also means in medicine, it turns out there are about 10,000 different diseases that are due to just one genetic mutation. So just one gene is wrong. So in theory, what you can do is you could just take that gene out or the part that's wrong and replace it with a good part. So all of those should be treatable in the next 10, 20 years using CRISPR. Here's one of the most famous diseases, sickle cell anemia. It turns out that African Americans have high incidence of sickle cell anemia because it's a disease that comes from Central Africa. And people who had sickle cell anemia weren't susceptible to malaria. But if both parents have the sickle cell gene, then you get sickle cell disease. And what happens in sickle cell is that the red blood cells, which are normally donut-shaped, when they get stressed, when there's not enough oxygen, they collapse and they become a sickle shape instead of a donut shape. And they can't carry oxygen. So people who have sickle cell disease, they run out of air very quickly, they get tired very quickly, the air doesn't get to their organs, so they can have organ failure, doesn't get to their brain, so they get headaches, and typically die much, much earlier than they should. Victoria Gray had sickle cell disease, and so badly that she would go to the hospital 7 to 10 times a year for kidney failure, liver failure, trouble breathing. it's one little amino acid that is wrong in the gene of hemoglobin. Hemoglobin is the part in the red blood cells that carries oxygen. So there's just one little amino acid that's wrong. Now, it turns out that when we have an embryo in uterus, the embryo has what's called fetal hemoglobin. It's a special form of blood that just the fetus has, and it's very good at transporting oxygen. When the baby is born, within about 4 months, that gene is turned off, and the baby no longer makes fetal hemoglobin, instead it makes adult hemoglobin. With Victoria Gray, what they've done is they've removed stem cells that are responsible for making blood from her spinal cord, taken them out in the lab, genetically modified them so that these stem cells, instead of making normal adult red blood cells, make fetal red blood cells. And then checked it that everything's okay, put those red blood cells back. And now, the stem cells back. Now those stem cells make fetal hemoglobin. So she is actually an adult walking around making baby hemoglobin. And it's been close to 2 years now, and she hasn't had one reason to go back to the hospital except to get her treatment. So it's worked really, really well. And 2 more patients now have joined this clinical trial. So you can see how potent this technique is. It works really, really well, especially when the genetic defect is in something that you can take out of the body and then put back in again. Or if it's in an organ that's isolated, and it turns out the eyes are very isolated too. You can, your eyeball has really just got a few attachments and the optic cord. And so it's not as dangerous as trying to genetically modify the brain or the heart or something like that. And so there are also some trials right now on humans trying to correct some genetic defects in eyes. So those are the 2 medical trials that are going on now. So what are the problems with this? Well, actually, let's just talk about some other uses. And that would be, of course, in food. You could now genetically modify food so it has more vitamins in it. You can genetically modify it so it's more efficient at photosynthesizing, more resistant to certain pests. There's somebody at UConn who has genetically modified grass so that the grass grows denser but shorter, so it doesn't have to be mowed all that often. And so you can do all these different genetic modifications fairly easily now. And some of these genetic modifications you could have got by breeding different types of grasses together, but others are completely different. So you can take a gene that you know works really well for a species that would never cross with grass. You could put it into the grass and the grass will adopt it. Now the rules in America are if it's a plant or vegetable or anything like that, agricultural, you don't need to label it, you don't need to get permission to do that. So anybody can do research on tomatoes, for example, new tomatoes that fall off the stem very easily. As soon as you want to genetically modify animals, that could be salmon, could be cows, then you have to go through the Food and Drug Administration, and then it becomes a lot more expensive and difficult to do it. So in America, at least, what we'll probably see is lots and lots of genetically modified plants, but not animals, because that's too expensive. We've probably all heard about the 2 set of twins that were born in China that had been genetically modified using CRISPR. So a Chinese researcher working in conjunction with an American biotech company genetically modified a set of twins so that these twins would be less likely to be infected by HIV/AIDS. Obviously, the twins couldn't give their consent and weren't asked about it. So genetically modifying unborn children certainly has some ethical implications that we should consider. The researcher who did this thought he was going to become very famous, but in the end actually got thrown into jail. He got a 3-year jail sentence because you're not allowed to do this in China. Had he done it in America, it would have been legal, as long as he wasn't using any federal funds. But the first people to come and talk to him were fertility clinics, especially from Saudi Arabia and from the UAE. And the reason they were very interested in this is they were thinking of genetically modifying in vitro embryos, not to prevent them get diseases, but to actually make them healthier, to make them better. It turns out there are a couple of thousand of genes where you can just change, again, one gene, and you can improve the endurance of a person. It turns out that endurance runners have this genetic gene that's quite rare. And if you go and look at the ones who've won the marathons, most of them have this genetic mutation, just naturally. But now you can have your unborn child genetically modify it so it has this high endurance. You can make stronger bones. You can have people that need less sleep. You can have people that don't have smelly underarms. Those are all single gene mutations. And so this is really a very scary thought that the rich people will, for the first time really in history with this kind of technology, will not only be able to have better lives, but they'll actually be able to become better because they will be able to genetically modify themselves, that they have more endurance, that they sleep less. And they can get system upgrades, just like your iPhone. Every 2 or 3 years, they can go and genetically have themselves modified. Yuval Noah Harari sort of says it's a bit like we're in charge of evolution, there's no doubt about that, but we're now creating a new species, Homo superior, which is going to be different from Homo sapiens, and it's going to create even bigger inequities in wealth. So that's another danger that we really have to think about when we're thinking about all this power that we have in genetically modifying things. And then maybe the biggest danger of all is something called a gene drive. So if you look at normal Mendelian inheritance, here you've got a blue fly and a red one. And this red one, let's say it's been genetically modified to be red. When the blue fly and the red fly mate, half the offspring will have the genes from the father and half from the mother. So this one's blue, this one's red. Then this red one breeds with the blue one again. Half of them will be red, half of them will be blue. The blues mate together, there's no red, so they all got blue offspring. So all of these are blue. So what you can see here is after a couple of generations, you've got mainly blue and very little red. A gene drive, it's like having a virus in your computer. What this does is what you put inside this red fruit fly now is a gene that makes it red, and the CRISPR, the gene editing, the word processor, you put that in there too, and you program it so that whenever it sees any other color besides red, it cuts it out and replaces it with red. So these two mate, you get a red and a blue one, but these two mate, this one actually would have given a blue one, but this sort of CRISPR bomb goes and changes the blue to red. And then the blue mates with the red mates with the blue, all its offspring that aren't red are cut out to make them red again. And very soon you see that all the offspring are red. There have been some calculations that if 3 years ago we released a fruit fly with, say, red eyes or yellow eyes and a gene drive, all the fruit flies in the world will have been changed. So why would you want to do this? Well, it turns out that there are some diseases like malaria. About 300 to 400,000 people die due to malaria a year, mostly children under 5, mostly in Africa. In fact, in just this little time that I have given this talk, about 20 children would have died from malaria. So could we go, and the answer is yes, to the Anopheles mosquito and genetically modify it so that when a male and female mate, the female lays all the eggs, the males all hatch from eggs, become larvae, the larvae all grow up to be males, but the females all get stuck at the larval stage. They never become mosquitoes. The males have this gene drive so that all their offspring will do the same thing. So all the males will always mate with a female. The female will lay eggs, but will never produce new females. All you'll get is males that make more males, and the females never get past the larva. Now, there's 2 reasons for doing this. One is it turns out that females that are pregnant are the only mosquitoes that bite. So it turns out that when you get bitten, it's always by a pregnant female. She's looking for the blood to feed her eggs, her offspring. Males actually eat pollen. So if you've genetically modified a male, the male is not going to bite you. So there's no danger of anything crazy happening there. And so you can collapse the whole population. So they've done experiments at Imperial College in London with Anopheles mosquitoes that have been genetically modified like this. And you can have 10 mosquitoes, you can release 10,000 wild type mosquitoes, and those 10 mosquitoes within a month will collapse the whole population of the 10,000 that you've released. So it's a very, very rapid way of introducing the gene drive. So now, for the first time ever in history, we have the power to wipe out a species very targetedly. And so we really are in charge of evolution now. Of course, you could put any genetic modification you wanted to into mosquitoes. Now, mosquitoes breed fast and quickly, so it's a lot easier to do this with mosquitoes than with humans, right? If you did this with humans, you've got to wait like 100 generations, which takes a long time with humans. But with mosquitoes, it doesn't take long to get that. But there, as you can imagine, there are a lot of dangers with something like a gene drive. So that's sort of the part I wanted to talk about, CRISPR, about genetic engineering. I think everybody can see the power of it with curing diseases, but also that the fears. Anybody have some comments or questions?
I have a question.
Yeah.
Is there any way, like, say they managed to get rid of the Anopheles mosquito? Is there any way to predict what kind of ricochet effect that would have on other species? The way, getting rid of wolves has affected the populations of. Is there like any kind of algorithms for that or whatever?
So there are 2 things that at least the proponents of gene drives would tell you. The one is that this is species specific, right? The Anopheles mosquito is not going to breed with any other type of mosquito. So that makes it really, really targeted. The other thing is that the Anopheles mosquito is an invader in most countries. So it wasn't there 500-600 years ago. We have brought it and traveled it around. It turns out that mosquitoes fly no less than a mile in their whole lives. So the only way they get around is by traveling with people, sitting on your back or in your car or in the plane, in the ship. But we moved them from this tiny little area in sort of Central Africa throughout the world. So the argument would be that there probably isn't anything going to happen, but there's probably another species of mosquito that will take that ecological niche. Maybe a more aggressive mosquito, but hopefully not a mosquito that carries malaria.
Okay, thanks.
Another problem with this is once you've sort of started it, it's going to be really hard to stop it, right? It's not like a pesticide where you can release the pesticide and you can say, oh, no, I don't like what this pesticide's doing. In this case, it's going to go through your population, and once you've started it, you're not going to stop it. There's somebody at MIT who's studying Lyme disease, and he's thinking of either genetically modifying ticks or mice, and is starting to do studies on small islands near Martha's Vineyard. It turns out that Martha's Vineyard and Nantucket have huge Lyme disease problems. And so to get Lyme disease, you need both the tick, mouse, and deer. And so if you can genetically modify the mouse or the tick so it doesn't carry Lyme disease, then you're in business. So this is something that's also quite close to home to all of us.
All right.
Mark, I have a question.
Yeah.
It's Michael. Hi, I
Hey.
enjoyed your talk. I guess I have two questions about CRISPR. One is you talked about the difference between regulation in China and the United States in terms of gene editing. Is there any work going on internationally in terms of policy and regulation? And then my second question, I'm going to ask them both so I can mute again, is in terms of things like climate change, you're a chemist, is there any way, like you've talked before about cow farts and how it contributes to, how they contribute to climate change. Is there any possibility of using CRISPR to somehow adjust climate change?
So the first question about regulation. So the regulation is really, really a hard thing. Because as soon as regulations are enforced in one place, everything just slips into another country. And so you get medical tourism, you get labs where I know George Bush at some stage stopped the use of stem cells in America. And you found all the researchers, the American researchers sort of moved over to Europe where they could use stem cells. And now they're slowly coming back. But as soon as one country starts putting in a regulation, another country will fill that gap. So it's really, really difficult to do that. So the scientists themselves have met a couple of times and have made some sort of rules that sort of said, it's unethical to genetically modify germ cells. These are cells that carry on into further generations. But it's just a question of time before somebody's going to do it. And so what you need is some sort of fear that it's the funding agencies, the publishing agencies, and the governments. They're the ones who've got to work together. And basically, if a scientist has done anything like that that goes over the bounds, then their work shouldn't be published again. They shouldn't be funded for anything. And they've got to be strict rules like that. Then something like this might stop. But the American government just can't get its act together because the Democrats and Republicans have different views about how things should be regulated. And so the consequence is there is no regulation. The regulation is just you can't use federal funds to do any of this. So America, in that sense, has amongst the weakest regulations.
I want to make a comment here, Mark.
Yeah.
I don't know how long, you mentioned about going to school in South Africa. I don't know how long you've been here in the United States, but you may recall that it was only about 20 or 25 years ago that just the very basic stuff of genetic engineering was highly debated. The question is like, where is that going to go? There were rules even passed in the city of Cambridge, Massachusetts, of all places, about that. And, whatever. But I would argue that you need a society where these things can be discussed openly. People make their best case for and against, and you can make an argument about stem cells or anything else. But having that society that is willing to discuss situations like this is where you may get some controls put on. The problem is the world isn't generally like that. Certainly not in China, where discourse is not particularly encouraged. Policy is set at a high level, and they go forward. I mean, we saw what happened to the folks that spoke up against COVID earlier this year. Late last year, early this year, they got shut down, made to recant. So my point is, it's going to happen. I mean, and it's not going to be because of something we do here or don't do here. It's because there are societies that there will be no checks and balances on a central power that thinks that this may be a good idea that will create an advantage for them. So time to live with it, because it's coming. I can guarantee it's money in the bank on that one.
Yeah, because of everybody wants to compete, everybody wants to be in front. And so it's going to be very, very difficult. Now, the Cold Harbor Springs discussions that occurred about 20 years ago, the problem is that CRISPR is so much more powerful and has come about so fast that all the sort of discussions that took place 20, 30 years ago, just don't hold water anymore, because CRISPR can do things. And in those days, things were aided by the fact that it was hard to do genetic modifications. Now it's just so easy.
So you're going to have a whole contingent of people trained in this technique that are going to be able to do it, and there will be absolutely no controls over it in societies that won't be able to pull funding or other disincentives for not following the rules. So it's going to happen, and it's going to be a freaking mess, unfortunately. Whether it comes in 10 years, 5 years, or 50 years, it's going to happen. I mean, unfortunately, science fiction is sometimes, predicts the future.
Yeah, and so in the front of all this is somebody called Josiah Zayner. He's a biohacker in California. He gives online courses on how to genetically modify yourself, and has been on YouTube with a tattoo gun, injecting himself with a virus to actually genetically modify himself. And you can buy the kits. It's about $1,000. Most people are using them to genetically modify dogs, to get bigger and stronger dogs. But he actually has a kit for genetically modifying humans. So let me show you the last technique. This is, to me, the most amazing of all of them. And this is deep learning. So in 2015, the best computer program, chess computer program, was a massive, massive computer that did 70 million calculations per second. So when it played against a human chess master, it basically worked out all the moves that the human could make and all the moves that it could make. I mean, think about it, 70 million moves. It had been programmed by humans who play chess. So it was just like a big, clunking, huge machine that could beat humans. And most of the time, it beat humans. In 2016, AlphaGo came around, and AlphaGo used what's called deep learning or machine learning. And basically, in 4 hours, what the programmers did, they gave these computers all the chess games that had been played between masters that were on record. And the computer then was told, these are the moves, and if you got the king in checkmate, you win. And with the algorithm, the computer taught itself how to play. So in deep learning, you give an algorithm lots and lots of data, and it looks for correlations between the data. Correlations that we can't see, and correlations the program doesn't even tell you that they exist. So after learning for 4 hours, AlphaGo, which does about 50,000 calculations in a second versus 70 million, beat the bigger computer. They played 100 games, and AlphaGo won 21 times, and 79 of the games were draws. But the big computer never ever won. Gary Kasparov, who on the cover of Science, wrote an editorial saying that the computer used moves that he, who had been a world champion in chess, had never ever seen before, used strategies that were totally new to him, and were actually quite beautiful. So, but chess, it turns out chess is what? 10 to the 50,000 different positions. So 10 with 50 zeros, that's a lot of different positions. Go is much, much more complicated. It's got 10 to 170 different positions. And so it's much harder. Well, it took the computer using machine learning less than 6 months to learn how to play Go. And AlphaGo beat the Chinese champion, who was the world champion Go player, and then the South Korean champion. It was watched by 400 million people. That's about 5 times as many people as watched the Super Bowl. They watched this computer beat the top player. And apparently, it was like a Sputnik moment in China. From that moment on, everybody was really excited about machine learning. And the Chinese went in gung-ho into machine learning. Now, if you remember, I told you that I'm a computational chemist. I calculate how molecules form certain shapes. There are probably about 5 to 10,000 computational chemists in the world. We've done PhDs, we've worked on these programs all our lives. We use mathematical equations based on physics and based on chemistry to calculate the shapes of molecules. Well, they took all the molecules of which we know the shapes, used machine learning, came up with a program that is more efficient at calculating how proteins fold and what proteins shape adopt than any of the programs that had been written ever before. Now, we don't know why it does it, but it can really predict the shape of a molecule better than any other program. So this is a really, really potent technique. Probably if you think about the everyday person, where you're going to see it next, probably in medicine. It turns out if you've got an MRI scan or a CAT scan, X-ray, mammogram, those are very easily read by a deep learning program, because if you think about it, what it's doing, it can have a memory of 10 million scans that have been done and the history of what happened with those growths and where they were. And so it can see things that the human eye wouldn't detect. It can see relationships that we wouldn't know about. And at the moment, it's about as efficient as a human, but millions of times faster. So the only reason it's not being used in America is just because of the medical associations are pretty strong. But if you went to go and have a CAT scan or something in China, I can assure you the computer would be doing it first and the human just checks afterwards. In rural China, if you go to have a checkup, very typically there's a computer that does a triage at first. So all the blood work, everything else goes into the computer. The computer then goes into the data bank of hundreds and hundreds of millions of patients. It looks at your blood work, compares that with the blood work and symptoms and everything else of yours with the hundreds of millions, and then finds relationships, and then says to the doctor, hey, this, and this is a possible disease. At the moment in America, Amazon and CVS are the two leaders that are working on this pretty frantically to try and catch up. But I wouldn't be surprised if in 10 or 15 years, we will all have a tiny little capsule floating around in our blood that is continuously monitoring a bunch of analytes, calcium, cholesterol, the temperature, hemoglobin, oxygen content. And all of this goes into a supercomputer, and the supercomputer then uses deep learning to find out what's wrong with you. And you'll get a message on your cell phone that says, hey, Mark, I think you'll be having a kidney stone in 3 days if you're not careful. Do this, do that, do this, take this medicine, and you won't get the kidney stone. So we'll have preventative medicine before a disease comes along. This means, of course, that doctors will have to be completely retrained on all kinds of things like that. But those are sort of areas in which deep learning is going to go to. And so if you ask yourself, I'm sure most of us have done that when we download an app, when we use Facebook, when you use Google, we've got this product, we're getting it for free. Why is Facebook free when people are putting all this effort into it? It's not really the ads. I mean, how many ads do you really see on Facebook? It's because they're collecting this data. And from this data, they can mine this and get all kinds of crazy information that we would never even think to see. Information that we might have something, some comment about something that we like. That might say something to this program that we don't even know about ourselves, but that the program can see from looking at hundreds of millions of people. Now, one of the things that Facebook did, for example, they started this craze where you would put a picture of yourself and a picture of yourself 10 or 15 years before. And everybody did this. And the reason Facebook was doing this is because they wanted age regression information. So they could have pictures of you at different times, and then they could use this program to figure out what you would look like in 10 years from now, and how somebody else would look 10 years from now. So there's all this information. So information has suddenly become really, really important. So if you go looking through history, what we see at one stage when agriculture was important, then land was really important. So countries that had the most land had the most agriculture, and they were the most strongest countries. Then the Industrial Revolution came through, and then having factories was really important. At some point, factories no longer became so important. At that point, science and innovation and technology became really important. And data might be the next one. So we sort of see all these different things becoming more important. Again, some of the sort of philosophers who are thinking about this are saying, well, if you had a capsule that's going around your body measuring all the analytes, that could also measure your oxygen content, your temperature, and by measuring that, really your emotions. And so what you would have is something floating around you, which could see when you are unhappy with something. And so you're responding to a political ad on TV with joy or revulsion. And so the company that's monitoring everything actually knows nearly your emotions and your thoughts. So that's one of the sort of scary ways that that could carry on. Just sort of end here, two quotes that I like. One of them is from Janet Doudna, and she's one of the two chemists that won the Nobel Prize earlier this month for the invention of CRISPR. And what she said was, what will we, a fractious species whose members can't even agree on much, choose to do with such awesome power, like the power to genetically engineer cells? And then I grew up in South Africa in a small town called Sasolburg. It was completely flat. There were just bushes all around. And as a kid, I loved to play in the veld, but I also liked to read about mountain climbers. Chris Bonington is an author who wrote about all these mountain climbers, and Reinhold Messner, another one. And George Mallory really fascinated me. So I started this talk with a quote about being the right size from 1926. This is from the New York Times, 1923. And George Mallory was a mountaineer who tried to climb up Everest a number of times. And the New York Times asked him, why do you climb up Everest? And he said, it's because it's there. So in 1924, he climbed Everest with her partner. They climbed up and disappeared. No one knew what happened to them. In 1999, they found his body about 100 meters from the top. We still don't know if he actually made it to the top and then died coming down, or if he died on the way up, but he nearly made it to the top. But I sort of wonder what he would say about climbing up something because it's there, if he knew that he was going to die, or perhaps if he knew that many years later, this is the spot pretty much where he died. It's just 100 meters from the top of Mount Everest, that really rich people go and get ported up the mountain, up Mount Everest. It's no longer a challenge, not much of a challenge anyway. I'm sure I couldn't do it, but to somebody who's really fit, and hordes of people go up, pay tens of thousands of dollars for the privilege of going up. And it sort of devalued the mountain. And so it's sort of my analogy of what's happening in science. Maybe there are some peaks in science, like genetically modify humans to get better humans, that should be untouched, that we should not go to. I don't know how we would do that, but that's sort of what I think about it. So that was sort of my final little thought. So if there are more questions, very happy to answer them.
Well, it's difficult to be optimistic that science will show restraint, because basically it never does. I mean, Mary Shelley tapped into that 150 years ago with Frankenstein, and very little has changed since then, I think. The scientists working on the atomic bomb knew that they were about to uncork a genie, and that happened, for better or worse. But science, as I put in the chat, hubris, thinking that we know things, thinking that we can control things, is really an occupational hazard of educated people. We always think we know more than we do, and sometimes we pay dearly for that position.
Yeah, no, totally agree with you. Oh, I see you wrote in chat, what about dinosaurs? George Church, maybe you were thinking of him. He's trying to genetically modify elephants with mammoth genes. So he's putting in all the genes from a mammoth into an elephant so that he gets a mammoth coming out there, or an elephantine version of a mammoth. Of course, in Jurassic Park, they were looking for the genetic information from amber. I don't know if that's possible yet. I don't think that's going to be possible for a while.
You know, I put that in very early when you were talking about the size of animals.
Ah,
And obviously, you know, the environment was somewhat different, but clearly you had a lot of very large animals running around for quite a few million years. And so, you know, that may sort of shoot that argument down. I don't know. It's not really germane. So, but anyway.
yeah, they might have, I'm not sure what the temperature, body temperature and things like that, yeah.
I've got one more question, and this is pretty theoretical too. If there was a sudden advance in genetically modifying food products, I guess you know, Roundup was a predecessor of that. Would there be like, say, a type of corn or type of wheat that would be so superior that it would take over whether we wanted to or not, and become a monoculture, the way you know, bananas has been inbred so much that, you know, every banana in the world is susceptible to the same disease? Would that be like a larger problem than it is now?
I presume so. Certainly if you used a gene drive, then that would definitely be true, but people wouldn't use a gene drive for something like that.
Okay.
So,
Let's hope not anyway.
right,
Unless you want to corner the, you know, the bread flour market.
right. So, but I think just with anything, right, if you've got something that's improved, more and more people will want it. And as more and more people want it, you'll get a monoculture, just like you described that.
Okay. Thank you.
Maybe so CRISPR is a genetic defense mechanism that bacteria have evolved themselves. So again, it's something that we've just appropriated, just like in optogenetics and fluorescent proteins, from nature. The first use of it was actually in yogurt and cheese, and they genetically modified the milk fermenting bacteria so that they wouldn't be attacked by other bacteria. And it turns out that if you eat cheese or if you eat yogurt, you've already had CRISPRized bacteria in your stomach, because there are billions of trillions of these milk bacteria that have been CRISPRized, that are in all the yogurt, organic or not, and in most mozzarella cheese.
That's reassuring.
Yeah, that doesn't bother me. So it's just interesting to know.
Yeah, it is. Had no idea.
Alrighty, well, it's 8 o'clock now. So I think we should wrap it up. Does anybody have one last question?
Thank you, Mark. Really enjoyed it. Thank you, library, for putting this on.
Yeah, thank you.
Thank you, Bethany.
Yeah, thank you guys for coming.
Thanks,
Yes,
Bethany.
thanks, Mark, so much.